leukemia. Basically, how does leukemia develop and what are the factors that determine which leukemia it is? In this slide, you see an overview of the bone marrow. And the bone marrow is actually the site where all the blood is produced. And the way you have to look at it is it's a very complex environment which is embedded in each bone. So here, it's not only the femur as shown here, it's also like in your vertebrae or in your bones in your head. So you can find it anywhere. So blood production is happening throughout the whole body. The output is very high. So per day, 500 billion blood cells are produced. So this means this is a highly active organ, and it needs a lot of factors contribute to the actual production. So for example, the output of production results in different cells. And when you look at this photo here of the bone marrow, you can see that we call it a very colorful picture. So you see different cells and different sizes with different shapes and different colors. And this is because the bone marrow is a very vibrant environment. And the way it works is everything is based around a hematopoietic stem cell. And you can imagine the bone marrow as like a beehive. You have a queen, and from this queen, all the workers derive. But once you lose the queen, or the queen leaves the hive, basically the whole tribe dies. And this is how it happens, and this is basically what happens in the bone marrow. So you have a hematopoietic stem cell, and from this cell we have progenitor cells, which are cells that are still primitive, but they're already committed to a certain lineage. Then you have all these progenitor cells, and at the end you get mature blood cells that appear in the peripheral blood and in the bone marrow. These are the cells that do the actual work, such as like supporting the immune system or blood clotting, or as for erythrocytes, carrying out oxygen. And this is very important to keep in mind because leukemia arises from this complex procedure. It's clear that this is a very simplified figure. It's actually more complex to how it actually works, and not all the steps are known so far. We do a lot of research, especially like Connie Eves does a lot of research on how hematopoietic stem cells work, how their output is regulated, but still there's lots of work to be done for that. When you look at AML, we learned within the last 30 or 20 to 30 years that acute myeloid leukemia develops from immature white blood cells. So at the very top of this picture, you have a hematopoietic stem cell, which is the most primitive cell. It's the queen of all hematopoietic cells. And from there, we have a common myeloid progenitor cell. So this is a cell which is already biased to produce either erythrocytes, platelets, granulocytes, or macrophages. And this is the white blood cell count we are measuring. And from these progenitors, more committed progenitors derive, and from them, mature blood cells arrive. So the way we imagine that leukemia arises is it's actually not always in the most primitive cell, because these cells are hidden in the bone marrow and we don't have a lot of them. And there's a reason why they're hidden, because they have to be protected from different influences. It's actually the more committed cells, the progenitor cells. And when these cells acquire some genetic defects, which we call mutations, or have undergo some other changes that eventually lead to a modification of how they work, what could happen is that they result in uncontrolled cell growth, which we call cell renewal, or which we call proliferation in scientific terms. So, and this uncontrolled cell growth, usually cells are regulated by itself. So they can grow, and then they die off at some point. It's like aging. But cancer is characterized by uncontrolled cell growth and proliferation, so these cancer cells can accumulate. But the other thing that happens is that these cells actually do not mature into white blood cells. So at the end, you end up with immature cells that grow but have no function. They just fill up the bone marrow and push aside all the healthy cells. So it's and this is a very nice picture here. So on the left-hand side, again, you see a bone marrow with a polymorph, meaning like it's consisting of different shapes, different cells, different colors. The nucleus are in different shapes. You can see like big cells, small cells, and so on. So it's a very vibrant environment, as I mentioned before. But when leukemia arises, you can see these monomorph blasts, and I've labeled them here. You can see blasts here, here, here. So they're filling up the bone marrow, and there are very few, for example, neutrophils, as you can see here or here. It's becoming monomorphic, and these cells take over, and this is basically what we define as, in this case, acute myeloid leukemia. And the original definition is if there are more than 20 or like 20% of blasts in the bone marrow, just by looking at them in the microscope, we call it acute myeloid leukemia. And based on these observations, so people try to classify